This is just basically putting in samples that are going to be analyzed. We've actually sequenced these samples already, about 16 samples. It's an 8-gene panel, AmpliSeq panel for orphan pediatric retinal diseases that we're sequencing the genes for. I'm going to select here a manifest, which we've preloaded. I'll click on here. I'll double check that we've got, we're using genome build 38. That's what our manifest, which is basically a description of all the PCR primers and their locations. And this will be run 1/2019. As I enter samples here, I'm selecting their index wells, and that just lets me very quickly fill in those and their index numbers. I can also see the sequences if we want. Once this is all filled in, I can basically queue this analysis, and it's going to run with these parameters. It's a read. Both strands, we're doing 151 reads. They both have an index read of 8. We have a germline variant caller. The variant caller depth filter we're going to leave at 10. We get a lot more depth than that anyway in this panel. BWA is the aligner that we'll use. Indel realignments are on. We're not using an imported example VCF file. That's optional. We don't need to do that. The annotations are going to be on the RefSeq part of the database, and once this runs and I requeue the analysis, it'll take about 110 minutes. It will align all the reads for all 16 samples to the human genome and look for variants that are different from the genome reference sequence and generate variant call files and reports that we can use to analyze sample data and do research to look for mutations known and new.